How does temperature affect the rate of cellular respiration in yeast? This is cellular respiration in yeast. We depend on decomposers to keep matter and energy moving through the food webs that make life possible. Yeasts are single-celled decomposers that are found in all biomes and ecosystems in the world. Yeasts depend on the environment for heat, unlike warm-blooded multicellular organisms whose bodies generate a consistent temperature. Whether an organism generates its own heat or it depends on the environment for heat, the organism needs a way to respond when the temperature changes in its environment in order to maintain homeostasis. We are going to explore the effect of temperature on the respiration rate of yeast growing in a sugar water solution by measuring the change in dissolved oxygen in the solution. The yeast cells undergo cellular respiration as they grow. During cellular respiration, the yeast cells will consume oxygen and sugar dissolved in the water and release carbon dioxide and water as waste products. Let's compare the rate of oxygen consumed by yeast when the external environment is at room temperature, when the environment is colder, and when the environment is warm and close to human body temperature. I have already connected the dissolved oxygen sensor and temperature link to my computer. This beaker has 200 milliliters of room temperature distilled water. This is another beaker with 200 milliliters of distilled water, which is cooling down in an ice bath. This third beaker is filled with 200 milliliters of distilled water that I'm going to let warm up to a temperature of about 35 degrees Celsius. For each beaker, first I'm going to measure the initial water temperature and also the dissolved oxygen conditions. And then I will add 15 milliliters of growing yeast suspension. Data collection will continue for 2 minutes. Now we're ready to test the respiration rate at room temperature. First, I will set the fast response temperature probe in the water, being careful not to touch the beaker itself, and I'm going to start collecting data in SparkView. If you're conducting this experiment live, you could see the temperature in the live data bar here in the corner in SparkView, but I'm going to start collecting data so it's easier for you to see the initial temperature in the video. Once the temperature is stabilized, I'm going to stop collecting data. And it looks stable, so I'm going to hit stop. I'll be telling you to record something when you need to write down results. So anytime you hear me say to record, you can pause the video and resume play when you're ready to continue. Record the water temperature for the room temperature condition in Table 1. Now I need to slowly stir the dissolved oxygen sensor for about 30 seconds to allow the sensor to stabilize. Next, I will measure about 15 milliliters, or as close as exact 15 milliliters of yeast suspension, pour it into the beaker, mix it, and collect data. First, I'll measure 15 milliliters at eye level. I'm going to start collecting data. Be ready to record the initial dissolved oxygen concentration in Table 1 as soon as I start collecting data. Here we go. Start. I'm going to add the yeast and continue stirring for 2 minutes of data collection, but we will speed up data collection for you. Record the final dissolved oxygen concentration and time elapsed in Table 1. I'm going to stop collecting data. You can see the time elapsed right below the digits display right here, next to where it says start. Let me rinse the dissolved oxygen sensor. And we'll repeat the same procedure with the cool water. I don't need to rinse the temperature sensor because it was only in water. So let's get the initial temperature of the cool water. Let me start collecting data for you so it's easier for you to see. And once the temperature stabilizes, I can hit stop. Let's wait for that temperature to get stable. I'm going to hit stop. Record the water temperature for the cold condition in Table 1. Now I'm going to perform the experiment exactly as the first run, and I'm going to speed through data collection for you. So first I need to slowly stir for 30 seconds here. I'm going to add the 15 milliliters of yeast suspension. I'm going to start collecting data. And you need to remember to record the initial dissolved oxygen concentration at the beginning of the run as soon as I start. That's good, actually. I'm going to start collecting data, record the initial oxygen concentration, Table 1, and here we go. There's 2 minutes. I'm going to stop collecting data. Record the final dissolved oxygen concentration in Table 1. Let's rinse and repeat with the warm water sample. Better put my sample back on the stir. Since this is a suspension, I've got to keep it stirring. Let's get the initial temperature for the warm water. I'm going to hit start to make it easier for you to see. And once the temperature stabilizes, oh, that's a bit warm. I need to cool this down, so I'm going to grab some ice. It's a bit too warm. I need it to be about 35 degrees. So let's add a little ice and see what that does for the temperature. You gotta wait for all of that ice to melt and make sure it's thoroughly stirred. Looks like we're gonna need another cube. That's perfect. Okay, let's wait for that temperature to stabilize. And that looks pretty stable. I'm gonna hit stop. Record the water temperature for the warm condition in Table 1. Now I'll perform the experiment the same as the other 2 runs, so I need to stir for about 30 seconds to let the sensor stabilize. I'm going to add 15 milliliters of yeast suspension. We'll start data collection. And remember, as soon as I start collecting data, you've got to record that initial dissolved oxygen concentration at the beginning of the run. Let me measure that 15 milliliters. Okay, I'm going to start data collection, get the initial dissolved oxygen concentration, and we'll speed through data collection for you. And 2 minutes have passed, so I'm going to stop collecting data now. Now that all 3 runs are complete, you're ready to complete the analysis. Refer to the end of the procedure for help with calculations needed to complete Table 1. Answer the questions at the end of the lab. While you work, think about this fun fact. I said before that yeasts are found in all biomes on Earth, and that includes the microbiomes inside your body and on your skin. Have fun!